Okay, so this will build off of some of the things that I talked about in my previous talk. I've tried to make it somewhat self-contained as well. And there'll be two parts. The first part will be about statistical genetics, and the second part will be about modeling, modeling things that can happen in cancer and what's called, we'll get to it. Okay. So classically, epistasis. Give me 1 second. I just want to move something a little bit so I can see things easier. Okay, great. Okay, so there's a classic idea of what's called epistasis, which is modeling interactions between genes or interactions between alleles at different loci. And there are kind of two constructions of it. So the first construction of it was by the plant biologist Bateson. And the idea that Bateson had was that some mutations stop other mutations, so they stand over them. And this is what he meant by epistasis. Now, the other version, which is going to be closer to what I'm going to talk about, is the idea of statistical epistasis. And this was something that R.A. Fisher coined, and he coined the term epistasy to mean any statistical deviation from an additive combination of two loci in terms of their effects on a phenotype. Right? So the question is, how do we get at these interaction effects? Okay. So let's talk about two basic problems in statistical genetics. One is associating genetic loci to phenotypes, and this was relevant to genetic architecture of complex disease and phenotypes. And then another problem related to this was what's called genomic selection, predicting phenotypic variation based on genotypic genetic variation. And this became very relevant to animal and crop breeding programs. So let's look at the data. Why is my phenotype? Could be crop yield, could be cholesterol level, right? And you have N of these observations. And then you have your genotype, which is N by P. And for simplicity in this talk, we're going to code up the genotypes as 0, 1, 2, which is basically just the number of minor alleles at each loci. So you have individuals and you have loci. Okay. Now, the classic model that people have used for genome-wide association studies and EQTL mapping is a linear model. And in some sense, the magnitude of BJ, of these beta J's, is evidence for association of locus J. And the sigma sub n will become important because, well, we'll see why it becomes important. And so if you look at this as a picture, right, this is what we have. We have our trait, we have our genotype, we have betas, which are effects, and then we have our variation. Okay, so what has been typically seen in animal breeding and plant breeding, and also some models of selective pressure in cancer, is nonlinear functions do better for genomic selection. So kernel Gaussian processes nowadays, neural networks, right? They do better. But the tricky thing to pull out of these models is what is the effect of a locus? Right. Ben, I showed you this picture earlier today. Okay, so there's this kind of trade-off between linear versus nonlinear modeling, right? So in high dimensional regression, smooth nonlinear functions are more predictive than linear functions. But variable selection is much easier for linear regression. The kind of analog of this, if you look at...
And we lost about half a minute since the beginning of the slide.
Okay, should I just start at the beginning?
Of the slide.
This one? Yeah, okay.
Yeah.
So there are two perspectives on talking about linear versus nonlinear functions. One is a statistical perspective, and one is a genomic perspective. In the statistical perspective, what has been observed is that high dimensional regression, smooth nonlinear functions are more predictive than linear functions in many applications. What else has become more clear is that variable selection is much easier for linear regression. So trying to figure out which variables are the most relevant is a lot easier for linear regression. In the genomic convention, what has been shown is using nonlinear models for genomic selection works well, right? So if you want to predict phenotypes, right, disease phenotypes, using nonlinear models tend to work well. But if you want to map the loci, if you want to do quantitative trait loci mapping, then linear models are way easier to use. Okay, so for two different applications, first for statistical genetics, and then for a cancer application, we're going to develop models based on very similar principles that allow us to do trait mapping and genomic selection. So these predict as well as the state of the art in genomic selection, and we have a scalable algorithm for trait mapping or variable selection. Okay, so two observations that kind of make what we're talking about possible is when you have the high dimensional setting where you have many more variables and observations. Again, like I said, smooth nonlinear functions predict more accurately. They also predict more accurately than very nonlinear functions, things that are very non-smooth. Now, what I will say, and something that has changed, is now with deep neural networks and massive data sets, when you are in the case where N is really big, so let's say P is, let's say, 1 million loci, and N is, 2 million people, this story is different. And nonlinear functions can do very well. Okay. And then point two, locally, a smooth nonlinear function can be reasonably approximated by a linear function. Okay, so let's start with a simpler example, right? This was an example that was popular for quite a while. And what is it? You have some function space, H script, and you minimize the loss of F to Y, you minimize your predictive square loss, and then you penalize these functions in a very particular way. Here it's what's called a reproducing kernel Hilbert space norm. Okay? And there's a very nice result, and the nice result tells us that this function f hat is going to be a linear combination of the kernel function. And there are many, again, going back to things like Gaussian processes, there are many really nice results in probability and approximation, which tell you that you can have an infinite dimensional space, but if you construct it nicely, you get back an n-dimensional problem. And here's your kernel matrix, and I'm just showing you what its elements are. And you can think of these X's. X1 is, let's say, the genotype of your first observation individual. X2 is a genotype of the second individual. Xn is the genotype of the nth or last individual. Okay, so what you can do with kernel methods, right, is you can expand and predict Y as a linear function of the kernel plus noise, right? What the problem that you have is trying to map this back to the original p-dimensional space like you would do with linear regression is problematic. It's not exactly obvious how to do it. So the classic idea of variable selection becomes a bit murky. Okay, so what we're going to look at is a Bayesian kernel regression method that provides an analog for the effect size for each exploratory variable in the original high dimensional space. So, like I told you before, the subspace of the functions that you can realize, you can rewrite as a linear combination of these kernel functions. There's an alternative parameterization where you can write the subspace in terms of the eigenvectors or eigenfunctions, really, of your kernel function, weighted by their eigenvalues, and then a linear combination of that. Okay, so these are two equivalent representations. Okay. So, and you can write the kernel function in terms of taking your eigenfunctions, weighting them by the square root of your eigenvalue, stacking all of those together, and basically multiplying them by the transpose. And this gives you a relation between the linear weights on these eigenfunctions and the alphas that are weighting the kernels. So this is all very classic. Now, the question is there a way to walk back and forth between something that's a linear representation and this kernel representation and this eigenfunction representation? So in theory, there's something that exists mathematically in the abstract, but the question is, how do you turn this into a statistical model or an algorithm? Okay. Another way of saying this is, there a reasonable way of getting a notion of a effect size or a beta out of these nonlinear models? So one idea is, can we project the nonlinear function onto the data? Now, if you just try to do this conceptually, right? And there's a reason why the second line is in quotes, is the dimensions don't quite add up. They don't quite work. Because beta is p-dimensional, and in theory, C is infinite dimensional, right? So the question is, what can you do and how can you adapt this? Now, there's a very nice idea that Recht and Ramini came up with, and is used actually in many contexts, and I'll talk about this in a second. So if you assume the kernel is shift invariant and it integrates to 1, you can rewrite kernel and expand it in terms of random Fourier coefficients. And this is called Bachner's theorem. Okay, so this is basically nothing but a Fourier transform of a kernel. And then you can write down the kernel, right, as an inner product between the Fourier coefficients. And that's just what we wrote down here. So the idea behind random Fourier features is that you pick these coefficients, you pick your frequencies, IID, you pick the offset, again, IID, and you just write down this transformation Z. So initially, this idea came from Recht and Rahimi, and their motivation to do this was to make kernel methods a lot faster and scale to large data sets. In the modern neural network literature, you'll also see this. And where you will see this is in something called the neural tangent kernel regime. So a lot of the theory of neural networks is driven by the following observation, which is if you have a deep neural network, you can well approximate it by a shallow one-level neural network. And there's a regime where you have a weight, and then you have an activation function. And the activation function, the weights within the activation function can just be random. And again, that comes back to this random Fourier expansion idea. Okay. So again, we can write this down just using linear algebra. Okay, so now we can project the nonlinear function onto the data by using this Z, because the dimensions now match. And so this gives us a way to walk back and forth between the kernel space, the basis representation, and the original p-dimensional space. So if we write down the generalized kernel model, yi is p as our density of y given a mean, where the mean is some transformation of this random Fourier expansion times linear coefficient c, then you can rewrite this in terms of an approximate kernel. Okay. K again is positive semi-definite, so it has this expansion. And so you can actually write this down as a factor model. And there's a huge amount of work on Bayesian factor models that you can just adapt for this. So what we can now define, again, just using linear algebra, is a general notion of an effect size, where your alpha, you can relate to the coefficients of the eigenfunction expansion. And then you have this relation to the theta. And so just again, using straight linear algebra, you can write down a notion of a beta, or a general notion of an effect size in terms of these C's. Okay, so now just to go through a Bayesian model, you have your model parameters theta, you have a prior, you have a likelihood, and what you're interested in doing is computing the posterior theta given your data D. And so it's just a prior times likelihood over the marginal likelihood. Basically, the denominator is marginalizing out the numerator. Okay. I'm sorry, wrong direction. So typically, we can't compute this posterior closed form, but we can sample from it. So we can give a state a hierarchical factor representation where you have these thetas and you have these y's, and U's are your random expansion. You model your prior on theta as a multivariate normal. Your model for the variance is chi-square. I'm sorry, your model for your theta is a chi-square. Your model for the epsilons are also chi-square. And this implicitly puts some prior, a normal prior on the betas. So we can also do this for 0, 1. So if y is 1, if si is greater than 0, y is 0, if si is greater than less than or equal to 0. So you can basically add an extra level and use this for categorical data. Same thing as I said before. Okay, so now there's one thing that is important when you start thinking about models in statistical genetics, which is the individuals may well be related, right? And this is especially true if you're looking at, let's say, mouse models for disease where the mice might be related, or there might be cage effects. This is certainly true when you're looking at studies of people that are related, right? This is certainly true in things like animal and crop breeding. And so this delta is what you model in terms of the relatedness, or what's called a kinship matrix, which is a way of quantifying how related two people are. Okay. So the way we get at this posterior is we basically can use Gibbs sampling. So we have our theta given the sigma of the theta and the sigma of the noise, and the data is multivariate normal. And because everything is multivariate normal, we know exactly what the mean and the variance are. And then we have a deterministic formula to get back our analog of an effect size, which is a beta. And then given that, we can get our variances. Okay, and this gives us MCMC samples of the betas. Okay, so now there are various things you can do. One thing you can do is you can do something called posterior probability of association analog, which is you can do a hard thresholding of these beta hats. And you can do a hard thresholding of these beta hats to get this gamma j. And the gamma j tells you whether you think that that variable is important or not. And that is your variable selection. Okay. So why might this work? Why might this make sense? And why might this get us towards some notion of getting interaction effects? Okay. So we'll do a warm-up example. Let's say we have a quadratic kernel. So kernel U comma V, these are p-dimensional vectors. Let's say they're the genotype at the loci, and it's just U transpose V plus some offset squared. Okay, so you can expand this. And if you look at the expansion, there's a linear term, right? That this is this square root of 2c UK square root of 2c VK. And then there's this higher order term on the left, which is quadratic. And then there's this other term, again, which is the interaction. So what you see in the second line in the top left is quadratic. What you see, second line on the right is an interaction term. And what you see just below that is your linear term. So if you look at a Gaussian kernel, you can look at a similar type of expansion. There's a linear part, and there are these two quadratic parts. And if you do a Taylor expansion of this, you get something which is a product over the peak coordinates. And so again, what I'm trying to convey is that these kernels can be thought about as these polynomial expansions with interaction terms. Okay. Now, that's fine, and that gives you something and some notion of the beta. But the question that often you have is, what is the combined pairwise interactions between a given variant and all other variants? Now, why might you want to know that, right? In the setup that I've given you an effect sample size. That's great, but I haven't given you a clear method of telling me how much one variant is interacting with all of the other variants, because that's hidden in this expansion. Okay. Now, and I talked about this in my previous talk. One idea you could try is to look at all pairwise interactions, all three-way interactions, all four-way interactions, right? And try to enumerate all of those. But that's going to be computationally prohibitive and brutal. Okay. So what you can do is you can define this marginal epistatic effect, okay? And how do you do that? You have a mean, that's your first term. You have a linear term, that's your xk beta k. And then you have this interaction between XL and BL. And then the last term before your noise is the interaction between XK and all of XL. Okay? And this XK beta K is the effect of the kth variant. So we can rewrite this in terms of the mean, the XK beta K, this MK, a GK, and an epsilon. I'm just rewriting all of these. Okay. So let's talk this through. XK beta K is the effect of the kth variant. MK is the combined additive effects of all other variants with the kth variant. Okay. And you can write again this down as a multivariate normal. And GK is what is a marginal epistatic effect between the kth variant and all other variants. And again, all of these can be written down in terms of a multivariate normal. Okay, so what does this do? This allows us for testing of marginal epistatic effects, which is a combined pairwise interaction between a given variant and all other variants. And the power of this is now we can test for this using p-tests rather than p-choose two tests. And this is this is a huge saving in terms of computation and statistical power. We were able to show this outperforms other methods on EQQL data, including Plink and two-step testing procedures. So there's another way of thinking about it. So if you do applied statistics, Gelman and Hill is a really good book. There's new version as well with David Dunson and Gelman and Hill. So you center the data, you center the approximate kernel, you orthogonalize the data, that's our spectral decomposition. You do variable selection. And that's in a way the philosophy. So I want to show you some results on real data. So the Wellcome Trust data for human genetics also has mouse data. And so this was from a while ago. So they didn't have a huge number of SNPs. They only had 10,000 and they had 129 traits. You can break that into behavior, diabetes, asthma, immunology, hematology, biochemistry. And we compared what we did to Bayesian lasso, Bayesian ridge regression, Bayesian linear mixed model, support vector machines. Again, we did 80-20 in and out sample, inner sample splits 100 times. And we looked at the accuracy of measure, which was mean square prediction error. And the initial point is our algorithm, Baker, did as well as a nonlinear method, which is the SVM. Now, why was that our first step? We wanted to show we were as good as a standard nonlinear method. And the results would have been the same for Gaussian process. Now, again, this is showing you the same plot, but across all of the different data sets. And what is interesting, and so lower is better, but what you find is that for behavior, it does better, but not crazy better. Diabetes, it does do better, especially for a few form. In asthma, it does better. Immunology, it does, again, somewhat better. And biochemistry and hematology, it does better. And there are certain cases where it does a lot better. And there's a question, why might that be true? For some phenotypes, why does it really make a difference? And other ones, it doesn't. Okay. So what we did to study that is we took a linear mixed model. Okay. We basically did a modeling of variance effect. We looked at the variance components of if we just have linear effects. We looked at the variance components if we have pairwise interactions. We looked at the variance component if you have third order interactions, it's in black. And we looked at variance component where you have the cage of the mouse makes a difference, right? And what you see that's very interesting is that, and let me just go back to the previous example, is that the cases where it really made a difference using these epistatic models is where you have pairwise third order interactions and common environment. So common environment, especially in diabetes, you see a lot, as well as in asthma and immunology. Now, what was also interesting and somewhat surprising, especially in immunology, is that there were third order effects, right? Which we didn't come in thinking that would be there. Okay, so we also looked at people. These are 14,000 cases, 7 common diseases, 3,000 shared controls, about 500,000 SNPs for each individual. And we looked at Crohn's disease and type 1 diabetes. And we looked at the posterior probability of association analog. And we were doing this for basically doing trait mapping, right? That also included epistatic effects. Okay, and so this is what's called a Manhattan plot. And we're looking at the posterior probability of association analog. And what I'm showing you is for Crohn's disease, right? In the blue are loci that were already known to be associated. And the red are new loci that we got by looking at epistatic effects. This is for Crohn's disease. And for type 1 diabetes, again, similar results. The red are ones that we found that went above and beyond the standard GWAS. Okay, so summaries and mapping, relatedness, local estimates of heritability, and we can make this bigger, faster, and hopefully better. And again, these are some of the papers related to this. Okay, now I'm going to spend about 15 minutes telling you a similar story, but in a different application, okay, which is myeloma therapeutic strategies that select against resistance by looking at MYC-driven evolutionary convergence. And let me explain all of that. So there's a drug. It's called verumafenib, and it targets a particular gene in the MAPK pathway called BRAF. And this is dealing with growth signaling. And so what can happen is A is a person before they got the drug, B is a person after they got the drug. But the problem with a lot of these types of medicine is that you get acquired resistance, right? And so if you look at this, right, if you look at the different disease stages, some people are very fortunate and they don't get resistant, but most people have this issue with resistance. And, you know, this holds over a few drugs. And what I'm showing you on the right is a Kaplan-Meier plot or a hazard plot of these individuals and their survival. Okay. So how do you get this resistance? How do you get this complex disease resistance, right? And for some tumors, the idea is they only develop through resistance through gatekeeper mutations. So BRCA-ABL CML is one example of this. This is, again, a myeloid leukemia. The problem that becomes more difficult is when your tumor can develop multiple resistance mechanisms. Okay, but if they all function through a clear pathway, then there's probably something you can still do. What becomes hard is if I look at the signaling pathway and the resistance is driven by multiple pathways. So you have multiple resistance pathways that co-occur within a single patient or a single tumor. An example of this is the case of BRAF mutant melanoma. So the question is, how do we get at that? How do we get at these notions of resistance in this more complicated case? Okay, and there are a lot of papers that talk about the RAS-MAPK pathway, the PI3 kinase AKT pathway, and FK beta B pathway, right? And the story here is it seems like resistance to this drug can be driven through many different pathways. So how can we get at that? And can we say, is there something that's underlying what's going on? And so this can become a game of whack-a-mole. Like if you hit one target, right, the signaling pathway goes down another. If you hit another, it goes down another. And so this becomes a problem, right? So how do we think about targeting it? Are there common effectors that we can target? Is there something that the drug can get at? So in the question of BRAF mutant melanoma, right, one may think that there might be a common effector, and that's MYC. But the question is, how do we know that, and how do we test that? Okay, so what am I showing you here? I'm showing you fold enrichment from drug to control. And if you see, MAPK has a change, PI3 kinase has a change, NFK B has a change, Notch1 and ER. So these are some really, really commonly known cancer driving genes. Okay, so what we did was the following. We tried to use a nonlinear variable selection method to try to pull out a signature. And what we were looking for is a signature where once you gave the drug, right, there was a difference. And then once you checked it later on, right, there wasn't a difference. So we're really looking for something which starts at the same point, changes, and then goes back down. Okay, so we didn't have really massive data sets here, right? So we really had to do more careful modeling than we could do really just brute force ML. So we had gene expression for 10 tumor samples with pre-treatment and on-treatment. Sorry, wrong way. And we had gene expression for 29 tumor samples with pre-treatment and post-treatment relapse. Okay, so these are the two small data sets we had. And our signature was what's differential between pre-treatment and on-treatment, right? And we intersected that with what's not differential between pre-treatment and relapse. Okay. So what I'm showing you now is in patients, the probability of MYC reactivation on relapse. So that's what I'm showing you on the left. And on the right is what I'm showing you are log 10 base factors for these different genes. Okay. And on the far right, you're looking at the difference between reactivated and deactivated. And so then, because my colleague Chris has a lab and does experiments, we looked at cell models. And we looked at the original is on the left, right? And we're looking at full change of MYC. And then in the middle is what happens when you hit it with this BRAF inhibitor, which is basically the analog of the drug. And then on the right, again, this is in the left panel, we're looking at what happens when it becomes resistant. Okay? And so to figure out whether we think MYC might be a checkpoint, right? We actually looked at MYC staining, pre, on, and post. And then what you see is pre, it's up. Once you get the drug, it goes down. And then post relapse, it goes back up again. Okay. And so we also looked at what the response signature genes are. So these are the top genes that are changing with respect to this signature, right? That there's differential pre to on treatment, and there's no difference pre-relapse. And these are the top genes for that. And what we can do is look at, for example, MYC, look at some other genes, and look at, you know, how you're seeing that difference. And then we also did, on those signatures, we did gene set enrichment analysis, meaning we looked at those signatures and asked whether they were significantly over or underexpressed as compared to, genes not in certain pathways. So if you look at the MYC pathway, MYC targets up. This is a given signaling pathway that we know something about. You can ask, are the genes in the signature significantly within that pathway or not? More so than random, right? And what you see here is a lot of signatures showing MYC signaling. So how did we do this? You've already seen this slide, but let me go through it for people who might not have been there before. If you are looking at statistics, there's this idea of smooth functions. Smooth nonlinear functions are more predictive than linear functions. Variable selection is easier for linear functions. I'll skip the genomics version. I showed this earlier, so we don't need this. We've got that. So, and I just showed you this. So we used exactly the same method. We use exactly that Bayesian kernel regression method to get at these interactions and to get at these nonlinear effect sizes for the betas. And what does this give us? It again give us a suggestion that there might be a common effector, right? Again, this is just what I showed you. So if I look at the genes, and if I look at pathways, right? You have HRAS, KRAS, MAPK, all of these pathways, right? And I'm giving you here false discovery rates on changes. So on the left, you see the sets that tell you what happens from getting the drug from what the changes that happen getting the drug. And on the right, I'm showing you the changes that happen on relapse. And if you look at the changes that happen on relapse, you see a lot of a mixed signature. And this is just showing you kind of a correlation plot of the similarities that you get. And so Chris's lab did more experiments to see if MYC is necessary and needed for resistance. And so we're showing fold change in fluorescence. And so you're showing first what's happening when you give BRAF. And then the LUC is the control, and the red is the MIC. And so you're starting to see what some of the differences are in MIC versus, in what's happening with MIC. And BRAF, MEC E, and ERK E, these are all different inhibitors, and it seemed to be affecting very similarly how things are going through in terms of the signaling pathway in MIC. And again, more experiments were done in various cell lines, right, to see if we get similar results across different cell lines. And the short answer is we were getting similar results across different cell lines. So the SHGFP is a control. The SHMIC is our MIC case. And what you see is that it goes down, but then it goes back up, right? Which is what's happening in vitro here. Again, another control. And this is actually what's happening in vivo. And that if you look at the change over days, right? If you can shut down MIC, right? Which this combination of DAB plus TRAM or DAB plus TRAM plus JQ1 can do, then you don't get this increase over time and this resistance. So you can in a way shut down the resistance. Okay. So you can also ask questions about, can you use synthetic lethality? Yeah.
And again, we lost internet for about half a minute, if you can. Yeah.
Here?
Yes, I think so.
Okay. So what I'm showing you here are in vitro and in vivo experiments, right, that were used to test whether having some control over shutting down MIC helps you with resistance, okay? And the top left, we're comparing GFP, which is a control, right, with just with a simple MIC inhibitor, okay? And we're showing you the picture, and what the picture says is that in this case, if you just try to shut it down with one MIC inhibitor, it doesn't quite work, okay? Because it's going back up again, and that's what you see in graph 10, okay? What we're showing you on the plot right below it, so this is the panel that is bottom left, is if you combine two different inhibitors, right, that is your black curve, right, you get much better results. And again, that's in vitro. So on the right is in vivo in mice. And what we're showing you is that if you combine these two inhibitors, DAB plus TRAM, plus DAB plus TRAM plus JQI, you do better. And having more drugs there, having a cocktail, works better than having one and having two. Okay? So now you can ask a question about synthetic lethality against resistance. So you have this MAPK sensitive and you have this MAPK resistance. And so there's a scale between BRAF and MIC. And the question is, which might be driving this more? And did I lose it? Yeah. Okay. So you can look at RNA sequencing, you can look at metabolomics, and you can look at genetic and pharmacological screening, and you can try to integrate this to try to discover synthetic lethal targets. And so if you block resistance through synthetic lethal targets, this is what you find. You find this dark point, right? Which is a particular combination, is doing kind of well in terms of the cell numbers, right? And then in the lower image, again, PLX 472 plus glucose plus glutamine is again doing well. So this combination of the drugs with particular deliveries and a particular amount of glucose and glutamine are can give you better control. And the reason why having glucose and glutamine and controlling the amount of that helps you is it has an impact on the what's the right word? It has an impact on the local environment of the target, and it has an impact in terms of energy. Okay. And so this is what I was trying to say in the previous slide. It's not just a drug, it's also a level of metabolism. And so we can kind of tell you something about how there's an interaction between the drug and the metabolism and how that impacts the effectiveness with respect to resistance from the drug. Okay, so last thing I'll say, right, is that, again, this is different signatures, and we look at the gene expression of these signatures. And one of the things that you start seeing is that you also have these metabolic signatures that are helping to drive, I guess, resistance to resistance. Okay, so roughly speaking, what one can do is make up pictures. And this is not something we computed experimentally. This was a summary from the graphs that I've been showing you, but this is possibly a form of how you can get at MYC resistance through drugs and metabolism. Okay, the lines that you see with the T's, those are inhibitory. The lines you see with the arrows are directions, and the inhibitory lines are kind of basically things you can use to try to knock things down. And with that, I will end. But the story that I was trying to tell you is that what's interesting is something that you can use for statistical genetics, you can then adapt to studying signaling pathways.
Yeah, that's a very interesting talk, Sayan. Especially the results are quite impressive. I'll go through the papers anyway. Any questions from the audience? Go ahead.
Hi, I have a very general question considering all three talks so far. So how does a non-linear spline-based regression model compares to Bayesian kernel regression method in the context that if for a biologist who is studying variations at different levels, how would they choose between a random effects based mixed effects model or a Bayesian model?
So this is a very good question. So you can actually write down a nonlinear random effects model. Okay. It's just that what you can't pull out of it is in the standard methodology, you can't pull an effect size out of it. And what we're giving you is a way of doing that. Number one. Number two, in terms of choosing between a kernel method and a Gaussian process method, let's say, for example, in a way, they're very, very similar in terms of the mean. It's just that the Gaussian process also gives you uncertainty. But in terms of the predictive part and the mean function, they're effectively the same.
Any more questions? So I just had a question about the, if you go back to the epistasis part of your talk and where you were showing those loci for these various diseases, diabetes and so on.
Or the humans or the mice?
I think it was human. Or
Okay.
I can't remember. Yeah. I think you've stopped slide sharing.
Oh, sorry.
Okay.
I will start again. Give me 1 sec. Is it back?
Yeah, those, yeah,
Okay.
similar ones. So the blue ones are the known ones and the red ones are the ones you predicted.
Yeah, so we recovered the blue ones, right? And the red ones are the extra
Okay.
ones that we got.
Okay,
by
so that was
adding- Yeah.
you recovered all of the blue ones.
Yeah.
Okay. And the red ones, are they validated in some way or? I mean,
We did not do the validation, no. So we don't know for sure. No.
right, because in this particular slide, there's a lot of red. So, I mean, but it's very impressive that you can do this. And okay, yeah, go ahead.
So there was another study where we had a data set of people in Naples, right? And it was a GWAS study, and these people were very closely related. And there one of my colleagues ran some of this stuff, and that was validated.
Okay, there's a question
And what
from Marushi.
what we found is that, and this is not probably surprising, is that when you have a lot of relatedness, modeling epistasis makes a bigger difference.
Okay, Arushi?
I'm sorry?
Oh, there's a question from back there.
Oh, okay.
Hello.
Yeah.
Sir, so you have said that this method gives effect size, right? So how can we interpret that effect size?
So there are a few ways of doing it, but one way we can do it, and what's more efficient, is this idea of marginal epistasis, right? Because the effect size might tell you something, but it doesn't tell you how much nonlinear effect you have.
And you have written that these effect size are the additive effect size. So can we interpret these beta coefficient as we do for the linear regression for each time point? Because these are additive ones.
So, okay, so there are two things you can do. You can get these notion of a nonlinear effect size, okay?
Mm-hmm.
But that doesn't necessarily tell you what the interaction effect is at a loci, right? And so the reason why we went on and did this marginal epistasis, and let me just add everything, is to be able to get at this last term, which is the interaction between, let's say, 1K and all the other L's, right? So to get at this marginal epistatic effect. And basically, we figured out a way of doing this as what's effectively either you can think of a linear mixed model or a glorified ANOVA.
Okay, thank you.
Anyone else? Okay, let's thank Sayan again for three very interesting talks.